Okay, welcome to the lecture on multiple sequence alignment. So last time we talked about pairwise alignment, and today we're going to talk about what happens when, like most of the time nowadays, you actually have lots and lots of sequences that you want to align. And not so surprisingly, that turns out to be a pretty tough computational question. But we're going to even take the next step. So in pairwise alignment, I took sort of one step away from the code and just talking about algorithms, talked about greedy algorithms, how they won't really work, and then recursive algorithms employed in the dynamic programming environment, or it's like an algorithm. The dynamic programming is an algorithm to efficiently compute recursive algorithms, so that you don't have to, you're guaranteed to get the optimal answer in the most efficient way. And so today we're going to talk about what happens when you've got lots of sequences to align, because the scoring mechanisms and things only really work for aligning two sequences at a time, at least the ones that we've talked about so far. So what do you do? Just to give a broad overview in terms of the workflow here and what you might be trying to do, just to provide some context. The first step is usually that you're going and getting some data from GenBank, or you've just sequenced a bunch of genomes or a specific gene. We're going to work on two gene families in this section to actually do multiple alignments, cytochrome P450 genes and 18S genes. But those are just two examples that I've pulled down that I had for my organism, and then a precompiled database on the 18S. But we'll talk more about how you get data, or how do you retrieve sequences for genes that you're interested in next week when we talk about BLAST. So getting sequences from GenBank, NCBI, that we'll talk about that next week. And then today we're going to talk about this middle step in actually doing the alignment. And then we'll get to a little bit about how to evaluate those alignments. And this can be an iterative process where you evaluate the alignment and then put it back into another aligner. At some point, you're satisfied that your alignment is good. I'm not sure why it keeps jumping back. And then you would go on and use that alignment to usually kind of address the questions that you came into the project really interested in. Something like phylogeny, how are these sequences aligned, or how are the organisms that contain these sequences, how have they evolved, how are they related to each other, what are the evolutionary forces that have acted on them. So looking to try to detect selection or something like that. That's what you would do after you've got good alignment. So actually the file name is data.zip. So that's the one that you'll work on. It's called data.tar.gz. Converted it. I'll actually change this before I upload the slides. But you'll want to get that from Canvas so that you can work on the homework set. And let me just show you that real quick. So in the module for alignment, there's data.tar.gz. You'll want to get that and download it. And we'll use that later on in the lecture. Okay. So once you've got your sequences, you will move on to then doing the multiple sequence alignment. And so I've listed three, really the three that are the most commonly used nowadays. We're actually going to use muscle. We'll do a homework assignment for muscle and MAFFT. I won't do Clustall. There are a couple different flavors of Clustalls. I know some labs in the biology department use Mega, which is a broader suite of programs that has Clustall built in. So we won't be using Clustal there, at least not today, but it is a commonly used software package for multiple sequence alignment. There are tons of other ones though, right? MAFFT, Muscle, Clustal, but there are a bunch of other ones that are some of them designed to act or produce better alignments on particular kinds of sequences. But you'll run into those if you're in that literature. You know, if, for example, it's particularly been good at aligning rapidly evolving genes, you might run into papers that have used a particular alignment software. But really, I mean, probably more than 90% of papers that I see, recent papers anyway, are using MAFFT if you've got lots of sequences, or Muscle, and then especially labs that are not heavy into bioinformatics tend to use Clustal because it has a nice graphical interface for it, and it's built into Mega and some of the other graphical packages. I think Genius might have Clustal built into it as well, or at least an easy interface for it. Just to give you an idea of how hard the multiple sequence problem is, back when we were programming algorithms, when we started to talk about counting k-mers, and especially counting k-mers with mismatches, we talked about the speed of our sliding window algorithm if we had to scan over the text multiple times. Well, you sort of scale that problem that I talked about at the beginning of pairwise alignment, where you had a known pattern with given distance and you're trying to align that to some sequence, and how that was a much harder problem if you had to align two large sequences and try to find the optimal set of matches. Now, when you expand that, it becomes something that scales, at least if you were taking sort of the brute force approach, as L to the n power, where L is the length of the text and n is the number of sequences that you have. So if you have a long gene and you want to align, you know, even 5 or 10, let alone 100 or 1,000, this is not doable. That quickly becomes a really huge number. Even heuristic methods, so heuristic methods are things that try to do it by a very, I mean, you can think of it as the same kind of 1-0 scoring that I introduced at the beginning of pairwise alignment, where you just get a rough score for how well two things align. You can get the algorithms to scale as the number of sequences that you have squared. But the awesome sort of innovations of MAFFT, Clustal, Omega, and Muscle falls in this camp too, is that they scale as the number of sequence times the log of the number of sequences, which makes them reasonably doable. So Muscle you can do on, you know, less than a few hundred sequences. So if you have 100 or less sequences, Muscle is really accurate and pretty fast. It may not be a significant improvement over MAFFT or Clustal, but the reviews that I've seen, so every time somebody publishes an advance in multiple sequence alignment algorithms, they usually do a comparison of everything that's come before, and Muscle often falls out near the top for accuracy, but not always for speed. And I'll show a graph in a minute. If you've got thousands of sequences, though, you are basically in the realm where only MAFFT and Clustal can handle it. And really, if you're in the 10,000 sequence realm, almost everyone is using MAFFT. Okay. The trade-off, and this is a trade-off that you just can't get away from, no matter how good your algorithm is you're always sacrificing the guaranteed accuracy with speed. You're having to take some heuristic. What I mean by that is some approximation of the best alignment, some distance measure, or you know, in pairwise alignment, we talked about the score. You have to take a simplified scoring metric that is really fast, and you're trying to optimize getting the best scoring metric, the best series of well, we'll talk about how you build a multiple sequence alignment, but the best approach to getting that optimal accuracy with how long does it take, right? It doesn't do you any good to be absolutely certain that you have the best alignment if it never finishes, okay? This just gives you an idea. This is mostly kind of for history and the development of these algorithms. You can see, you know, MAFFT takes multiple approaches, or I should say there are multiple approaches available in MAFFT. Some of them, right, these two falling over in the muscle family are a little more accurate, but a little slower. And then you've got this branch over here that are much faster, but perhaps a little less accurate. And when I say less accurate, it's not that they're inaccurate. And in some cases, you know, we'll talk about how to evaluate alignments toward the end today. But, you know, they're, in most cases, they're accurate enough that the speed makes it worth it, because you can always rerun the alignment after you say change gap penalties or something like that, if you're not satisfied, or if you do some of the quality assessments that we talk about at the end and it just doesn't look good. You could also just put in different regions of the sequences that you're looking at. Okay. So we're going to talk about these major advances. So what are progressive, iterative, and consistency methods? Mostly we'll talk about progressive and iterative approaches, and how, you know, the advances in these three things are kind of what make the branches in this tree, but it's not super important. You can go read the review or any of the recent reviews if you're really interested in development of multiple sequence alignment programs. Most of the time, most of your labs, if you're in the PhD program, are really only interested in get the alignment, okay? And so I'm telling you, you're going to use muscle or MAFFT or Clustall. This, what I was talking about in terms of the scalability. So on the x-axis here has the number of sequences that you're trying to align, and then how long does it take, right? So you get up here in the 3,000, 10,000. Muscle is doing pretty well, okay? And the circles are telling you average sizes, right? So muscle is taking a little bit slower time. So all those green dots. Now the other green ones down here are just a different parameter. So a different, well, you'll see in a second, right? So here they're taking a different distance measure in the first round of iterations of alignment, which doesn't really make sense yet, but you'll see in a second. And then you can use a Kamer-based method, which makes muscle much faster. Clustall Omega is sort of right in the middle. It is often used. And then MAFFT, the other one we're going to use, so it's lower down. And right, the thing to notice about this graph is this is mostly about speed and how that scales with the number of sequences. So the fact that MAFFT, this is why it's so commonly used when you've got lots of sequences, is it's really fast. So the blue dots are almost always at the lower end of the time scale relative to the other metrics. And you're really not sacrificing much in terms of the accuracy. Now, accuracy is not on this graphic, but I give you a citation there if you're interested. This was from the Clustall Omega paper. Just to give you some idea, though, about the accuracy. So higher accuracy here is better. Okay. So MAFFT, right? It's scoring really well in accuracy, and in this case, the highest. I should also say, right? So consistency, iterative, and progressive. These are different settings for MAFFT. And muscle too, for that matter. Just different models that it uses, different algorithms. Okay, so MAFFT scores really high on accuracy, and it's almost always the fastest. Right here is your basically your sort of exception where muscle was barely faster, but it wasn't as accurate when you made it run on its fastest mode. So how does this work? I'm basically just going to show you progressive and iterative because these are the sort of foundational elements for both muscle and MAFFT and what sort of makes them fast. The first step in a progressive alignment is to use to build a guide tree that is based on just some metric of similarity between sequences. So one way to do that is to make a really fast, simple pairwise alignment between all possible combinations. Now, that is not very fast, okay? But it seems like the most logical way to start, right? Let's run a fast pairwise alignment, right, with a simple scoring matrix. And then we will get this big matrix of sequences, you know, A, B, C, D, E, F, and what is their score when they were aligned to all the rest of the sequences. And then you can start doing, basically that gives you this neighbor joining, or I forget what UPGMA is an acronym for, but these are both using that matrix of overall similarity between the sequences to say, okay, align these two things first because they are the most similar to each other in this 4-sequence example. And these two are more similar to each other. So let's do an alignment between those two first. Okay, so you do that alignment, and this is the progressive. Okay, you do that alignment, and then you sort of take these two sets of alignments and build a consensus for those. So you build the optimal score, treating these like columns, okay? People recognized pretty early on that in some ways well, not in some ways, that progressiveness is a greedy algorithm, and it is really heavily dependent on that first step where you do the first fast analysis to get your distance metric, and then, right, that is the foundation. If that distance metric doesn't reflect or isn't a good reflection of which pairs you ought to align, early mistakes will be compounded later in the assembly. So if you have early mistakes here, right, you've got this indel in the wrong place then, and that is going to have implications as you sort of work backward into the larger and larger alignment. Okay, so strictly progressive algorithms, they're making that local optimal choice, which what that means is that first choice of which ones to align, and you're hoping that you aren't making mistakes, right? So making those locally optimal choices, you hope that they find the global optimum, but we know that that's not guaranteed to happen, right? Just sort of philosophically, greedy algorithms have that problem where you make a early mistake and that mistake gets compounded. So the next step in multiple sequence alignment progression, what made them better, was using iterative and consistency methods. So they start with a progressive alignment, but then they take different strategies on how do you evaluate that first set of alignments, and then basically rerunning that's the iterative part of rerun the progressive alignment based on some modification, and that whatever that modification is varies between Clustall, muscle, and the different flavors of MAFFT, the different models in MAFFT. And the thing that then sort of the last thing is to speed them up, like make the tree that first step of generating the tree that is going to guide your alignments, making that faster. And then faster methods for checking which parts of the tree need to be iterated and evaluating the consistency. Now, let's make some more sense out of that by just walking through what muscle does as an example. So the first stage of muscle is to build a progressive alignment, okay? Just like I was explaining before, you first get some similarity measure, the difference between the fast and slow muscle. Like the default muscle is fast and it uses a Kamer counting. So it doesn't even do that sort of fast pairwise alignment to get distances. It just does a sliding window, just like we did in the algorithm section, slides across the sequences, well, all of the sequences, and builds that Kamer library. The size of the Kamer library you can modify, right? If you have more closely related things, you might to do larger Kamers to give you a well, too many caveats to go down that rabbit hole. You can modify the Kamer counting, but in any case, you're not actually doing a fast pairwise alignment to begin with. You're just counting the Kamer distributions, and then the ones that you're going to move into that first stage of pairwise alignment are the sequences that have the most similar distribution of Kamers. So that Kamer dictionary that you built, which one is the most similar? And you know, based on the algorithms that you guys built in the algorithm section, that Kamer counting is really fast. Okay, this is not accounting. This isn't even the sort of final stage that we got to where you had to count it with mismatches. This is purely just slide along and make a dictionary out of all the Kamers that are there. Okay, and then count their frequencies. Okay, but you don't have to allow for mismatches or anything like that. You're just trying to see which dictionary is most similar. Right, so then you have your distance matrix. You know, you can envision this. You compare your Kamer counts from one sequence to another sequence, and you could think of it as, I don't actually know how muscle differentiates them, but you could think you might say, what's the value of each Kamer, and sort of total up the difference between the two values. So sequence A has, you know, ACGT, ACGT as a Kamer, and it occurs 5 times. And then you look at the other sequence, well, for that Kamer, how often does it occur? You take the difference of those two things, and that's a measure of distance. And you do that for all the Kamers that exist, total that up, and that's your distance matrix. That is really fast to compute. Okay. And that distance matrix, now you've done that comparison for all your sequences, that establishes the sort of tree, right? This thing that tells you what order you're going to have to align those things together. Okay, so that's your tree, the tree construction, and then making your progressive alignment. Okay, so then the sort of progression, the iterative and consistency parts of muscle is that it takes a second. So based on your multiple sequence, so now we have an alignment for all of our sequences. we can now go back and redo our progressive alignment, but in the first step, we have a much better indicator for what's the similarity between sequences because we've done an alignment already. So now you just take that alignment that you just did on the first pass and say, what's the distance? Like, what's my score for the distance between all these two sequences? And that now informs a new tree reconstruction. So now you've got a new guide tree, and it has this step of tree comparison. So, for example, on the first round, the guide tree looks like this, okay? Then you get, okay, what's the similarity between all these pairs of sequences? Recompute the guide tree based on the new distance matrix, right? Because this was the guide tree on the first round. Get the distances based on the alignment. Based on those new distances, recompute the guide tree. In this case, the guide tree says that X and V are actually, W and V are more closely related to each other than X is. So this section of the tree actually has some different patterns of similarity, like these sequences are more similar than just counting the k-mers said that they were. Hopefully that makes sense. So this tree is based on that first alignment. This tree was based on the k-mer counting. And so this is the consistency metric that muscle is going to use. That way, right, because this section up here, this tree, even though it's out of order, is still saying T and S are more closely related to each other than they are to U. So we don't actually have to recompute this part of the tree. And so that's a huge speed up in our algorithm, and that muscle only has to recompute the stuff where the tree changed. Right? Because you're right, if you redid this alignment, it should come out the same because the order of pairwise combinations is the same, but down here it changed, so your alignment is going to change. And then the last thing is use that new guide tree where you did not have consistency, right? So this part, you're going to redo a progressive alignment only on the inconsistent parts of the tree. And then you move on to stage 3, okay? And this is where muscle will break up the tree. And so it's basically subsampling different parts of the tree just to see if it stays consistent. So it will break the tree. The profile is, if you think back to this, it's going to build a new scoring matrix based on the best overall alignment. So right, this one, and you'll have to, if you don't remember scoring matrices, talked about those in pairwise alignment. But this is kind of how I talked about how the amino acid, the PAM and the blow sum scoring matrices were developed, right? You have a multiple alignment, and then the consistency here of L says that we should weight a change in this position, right? That should be really negative because it doesn't happen very often. Okay, and for example, on this position, you might say, well, I don't want to give this very much weight if it's a difference between W and F because it seems to be pretty tolerant of those changes at that position. Okay, so you develop that scoring matrix for your alignment that is based on the data that you have. So you do that partitioning of the guide tree, do the profile extraction that gives you a scoring matrix. You throw away some of the stuff that, right, where we have indels or regions where you can't really make a good alignment. Do the realignment, and then you iterate this. Okay, so you say accept it if you've recomputed this and there was no change in the alignment, you've done as well as you can, and then the algorithm should quit. Okay, and you're sort of working across the whole tree that way. So this is just a cartoon that kind of walks through that whole thing, right? Won't belabor it, but it just gives a few more details, like, for example, when it's doing iterative distance matrices to inform that first guide progressive. So the first one is counting Kmers to get that distance matrix. The Kimura distance is just the transition transversions having different weights. So obviously this is talking about nucleotide assembly, alignment, not assembly. Okay, and then you've got your trees, so you're iterating through there, and then in step 3, you start to break the tree apart and compute the subtrees until you can't get any better. Okay, and by better meaning the score is better. So first part of the homework is the homework's going to be due on next Tuesday. Okay, and we're going to align the tribolium, that's the flower beetle that my lab works on, specifically Tribolium castaneum, which is the model system, the P450 genes. This is something, I didn't pull this out at random. Last October, P450 genes are important across eukaryotes. Okay, in lots of insects, they're important because they can be related to pesticide resistance, right? Some of these are detoxifying enzymes, but they have lots of roles, and these families of P450 genes have been really hyper expanded in some organisms. There are, I think, 120 something of them in the data that we're going to use today. So this is the stage that we're at, and we're going to use the muscle aligner. How are we going to do that? Well, this is the breakdown, and we're going to actually work through that in class today, but just quickly go over it. The data.tar.gz file, I showed you that in Kansas back at the beginning, right? That's this one. Okay, so you'll want to download that. You also need to go to the muscle website. Let's open that link. That'll take you here, and you can go to downloads, and it says there use the Linux 64-bit version, so Linux and then 64-bit version. So you click on that and you'll download. This is another tar.gz file, and so you'll have both of those downloaded. And then move both of those, the data file and the muscle files, to Bridges 2. Okay, you should know how to do that on Globus by now. So I'm going to leave that part to, I'll go over it in class, but hopefully you know how to do that by now. Then you're going to want to use the tar.gz files and extract those. Remember, way back to our beginning bioinformatics tutorials, right? It's going to be something like tar-xvf, and then tell it which file, and it should extract it. You'll want to check the muscle manual has some notes, right? So the manual is here in documentation. So I've given arrows here, right? Installing it, it's very simple. One of the reasons that I even bother to do a muscle documentation, besides the fact that it's a good aligner, if you've got 10, 20, 50 sequences, muscle is really good. And what I'm going to show you today is often all you need to do to get an alignment. So installing muscle is easy. And especially because we're running it on Bridges, it's not always easy to get software running on the supercomputers. We'll talk about that next week with BLAST, and how they have to build BLAST specifically for that environment. Muscle is not that case, so we should be able to use it in a pretty straightforward way, and probably why some people still use muscle instead of MAFFT when they have a seems silly to call it limited, but you know, when you're on the order of 50 sequences. We used muscle even on, you know, tens of thousands of sequences with the gene family evolution stuff that I did in the mammals back in the day. That was before MAFFT, but it was still doable. So all that is to say, I'm not teaching you an outdated software set here. Muscle is still super useful. Okay, so you've got muscle and your data in your home directory on bridges. You're going to start an interact session, right? We had an example of that last week. So just type interact and wait for the node to open up, and then follow the instructions on the muscle tutorial. And we're going to run it all in the default setting, so that is super simple. Again, we'll work through that in class. Then for the homework, right, how do you complete the homework? You're going to have to get your alignment output back to your computer, right? Use Globus to bring that back from bridges onto your computer and then upload it to Canvas. All right. Now, that's it for part one of the homework. Once you've got your alignment file, that's what I want to see, that you were able to get that job run with output on bridges 2. Now, if this were some gene family that you were particularly interested in, you would definitely want to look at them. And so I've given you some pointers to software or platforms that you can use to upload your alignment file and look at or visualize what those alignments look like. Okay. If we have time, we may look at that in class, but I'm not going to spend time in the lecture video. You can do that on your own if you're interested, especially if you already have Mega or one of these other software platforms on Mac or Windows. You should be able to just open up the alignment file with those programs and look at it. So the next thing I want to talk about is MAFFT. So what happens if we actually have thousands of sequences? And I will walk through this in class, but I'm not going to do it here on the video because I've provided a really step-by-step guide for how to use MAFFT. And this is if you had several hundred, 1,000, 10,000 sequences, I would almost, well, not almost, I would always advise people use MAFFT at this point because it's really accurate and really fast. Right, so a bunch of slides here, really just walking through. There's no reason for me to be in that mode. So this is pointing where to click. We're going to use the online version. I've given you the link to the specific portal because MAFFT is available at multiple sites. The European Bioinformatics Institute has one. We're actually using the one in Japan where it originated, and it has the I think it has the biggest compute backend that's available via web browser. So here I'm highlighting all basically point and click. Should be able to walk you through aligning. In this case, we're going to use the 18S file to do the alignment, not the P450s. So that should already be in your downloads folder. You may need to extract that on your local computer. Okay. If not, you have it extracted on bridges too, so maybe bring that back versus via Globus if you don't want to do the tar command on your local computer, but you should be able to do that pretty easily. Okay, and so this just walks through doing the alignment on the MAFFT server for 18S. Okay. Just a note here. Sometimes I can't fit the whole web page on one thing. So this is like the top of the page, and then you've got dot, dot, dot. This is down toward the bottom of the page. Okay. So once you've done that, you've sort of done a MAFFT alignment. Then the step that we haven't talked about, and we're not really doing for the muscle, the P450 alignments, but this 18S data set has some interesting things in it that are good to illustrate in terms of how you evaluate your alignment. So when MAFFT finishes, and it may take a while, because you're gonna, at the end here, you're gonna hit submit, and then it may take a while. So you can give it your email address if you don't want to keep the browser window open, and it will email you when you're done with a link to your results page. So once you get here, first thing is you'll want to look at the dot plots that it makes with last. And so this is an example of perfect alignment, right? So these two sequences align perfectly and they're in the same orientation. You may see things like this, where this is representing an indel, right? This GA is not present down here, okay? But they still align pretty well and in the right orientation. Here, this is something you might see if there's been an inversion, because this is saying this piece of this sequence, it aligns well with this sequence here, but in reverse orientation. Okay, so this is matching on the same strand, this is matching on the opposite strand. It's kind of nice that last, the program that's making this, will check for this kind of thing, because lots of alignment programs do not, right? So if you have sequences with lots of rearrangements, you probably just end up with really bad alignments. And so MAFFT actually has this built in to show you what might be behind a bad alignment. So, right, go through those dot plots, look at what's wrong with the sequences. We'll ask these questions in class today, but I don't want you to have to, partly in the video, right, I didn't want to have everything pre-baked, just so there's some things to learn here in terms of this being something that we have to do all the time nowadays, and how you evaluate your sequences. Right. So once all of that is done, right, you've got your alignments, you've looked at them, if needed, you've made an adjustment. You can get your main results, right? So I think this actually comes in the email, right? So the last plots, right? This is the link, and then we're basically just moving down that results page. I can't remember if this is in the email or not, but anyway, it'll take you to a page where it should have both of these. So looking at the alignments will take you to this page, and so on, right? This will give you can download your alignments in Clustall format or FASTA format. Clustall format is putting so each sequence is on its own row, and then it's essentially looking at the text here, you can see the alignment. Okay, if you get it in FASTA format, it's going to have the species with its whole sequence and then the next species with its whole sequence, so you don't actually see the alignment per se, even though it still has all the dashes stuck in there. So the sequence itself between Clustall format and FASTA format is not different. It's just the way they're arranged in the file. Okay. Do you put the nucleotides next to each other, or do you put all the sequence from a given species together in the file. So one of the things you might want to do is to look at the tree. Okay, so just more visualization. And so here you can start to see some of the last things if you've gotten this far. So those plots tell you something about the orientation, and they're grayed out because they're not all the same length. So answers to some of those questions that I had posed earlier. You'll see some of these warnings. Again, I'm not going to walk through each of these. You've got basically a step-by-step, okay? And we will walk through this in class time, but you should be able to follow this even if you aren't there for some reason. Okay, so realigning based on trying to fix some of the problems with those sequences. Okay. Once you make the corrections to the data or limit the analysis, you should not get warnings when you try to look at the tree. Okay. And then that's an iterative problem. So you basically just saw MAFFT makes it easy to evaluate and then rerun. You might evaluate again and rerun. In this case, modifying which sequences are included or which orientation the sequences are in, stuff like that, in order to get the best alignment. Then the last step, and this is still, MAFFT makes it easy to move to outside software. It has a button to take this over to guidance, okay? And guidance is going to compute the confidence in each of the positions. Okay, so a little background on guidance. Won't sit here and read that to you, but guidance can take a while. And so this is not something that will run in class, but you can upload your alignment here. It only takes the FASTA format. Okay, so you'll have to get the FASTA format from back here in your results page, and then you can send that to guidance. And what guidance outputs is something like this that tells you position by position, how confident are you? So this stuff at the bottom are scaled to how confident you are, where a 1 is 100% confident. And what guidance is doing, this is showing not for the 18S, but for HIV alignments with SIV, it recomputes the alignment, leaving out subsets of the data. So if you took out, say, the HIV group M sequences and realign everything else, and then estimate, the conservation of sites, and then redo that for lots of different subsets of the data, dropping them out, that's bootstrapping. How often do you get the same alignment at each one of the positions? And that's what the blue bars at the bottom are telling you. And the nucleotides are color coded based on the changes or the confidence. Well, and the confidence. So darker red, for example, is more conserved. Blue is a change relative to the most common, and then the darker the blue is, the more rare that change is. This is a part of the 18S, so you'll get something that looks like this. The 18S ends up being really conserved in some right, the functional parts. So that's guidance. The last one is G blocks, and this is one where there's no automatic button to take it from MAFFT server to G blocks. So what you'll have to do, again, if you did this for Yeah, so you actually, that's a good question. I think clicking this guidance button carries the FASTA file over here, so you don't actually have to download the FASTA file, but to go to gblocks, you will. So download the FASTA file and then upload it to the gblocks server. And these are protein sequences by default. So you'll have to change that to DNA because your 18S is DNA sequence. Okay. And then just get the blocks. It'll take a little bit of time to run, but you should get an output that looks something like this. Okay. Technically the G blocks was designed to find highly conserved regions so that you could use it in phylogeny where all the data was present. So the underline in dark blue here is showing that there's no missing data and it has good alignment. And so in principle, those are the sites that you would want to keep, the ones that are underlined by these good blocks. Okay, if you want to retrieve concatenate alignment of sites without gaps, so resulting alignment, you'll want to pull that, and basically that's going to give you those sections that were underlined in blue. That's your G block result, and that is the part 2 assignment, is to get those G block results and upload to Canvas. So 2 files are going to go to Canvas. The alignment from muscle on the P450 genes and the G blocks output from the 18S. You'll upload both of those to Canvas by next Tuesday. All right. So just the last comment. So basically we've done all of multiple alignment, except, what do you do with that downstream? Okay, and that's another course. So molecular evolution, systematics courses, what do you do with your multiple alignment? That's where you're making evolutionary inferences or diagnoses based on your multiple alignment. Just going back to that paper on P450s, last slide here, just to show you, this is how it gets done and how it gets reported. So this P450 gene family evolution study used the online version of MAFFT. They just got all the P450 and CYP genes they could get out of the database using BLAST or annotation, because lots of them will have already been annotated, but some of them might be missed in the new genomes that are being posted every day. So they just got as many P450 genes as they could find and then aligned them with MAFFT. And then the rest of this that I didn't sort of highlight are the details of which softwares they used for the actual phylogenetic analysis, but they also did that part on Cypress, which is one of the Exceed resources that we don't have an allocation for because we're not, it's mainly it has all the phylogenetic software, so it's optimized for phylogenetic analysis, but since we're not going to actually get into how to do phylogenetic analysis, we didn't get an allocation on Cypress, but works very similar to what we're doing with muscle or MAFFT. Okay, just basically this is here to show you this is what you just learned to do in this lecture is exactly what you might like to do for a study of gene family distribution in arthropods or whatever your organism of interest is. All right, that's it for today. I will see you in class, and we will actually be doing the hands-on part of walking through that muscle and MAFFT exercises. Okay.